So, talking about that whole process, essentially we covered some of this. This is the process of gastrulation, and we'll focus a little bit on more of the differences between protostomes and deuterostomes and figure out which one of those we qualify for. So, gastrulation is simply the process where a zygote becomes a embryo, where the embryo becomes a morula, into the blastula, and then that blastula is going to start developing and making body tubes, because we are not just a mass of cells. There are tubes that run through us. There are hollow spaces where no cells are. There are body cavities that contain our organs. So we have to be able to make those tubes, and that tube happens like essentially our digestive tract, for us at least, happens. It's one of the first things that happens to the blastula in the process of development, called gastrulation. So imagine like a soccer ball or some type of other flimsy ball that doesn't have any air in it. It's kind of deflated. Now take that ball, and what I want you to do is imagine pushing your thumbs into it. It's kind of like you're making a pour into it. You're pushing a spot, an indention into the ball. Now imagine if you were to push that indention all the way through to the other side to make a tube straight through that soccer ball. That is the process of gastrulation. During this process, you are differentiating the cells by creating cells that are now on the outside, cells that make up the tube that go through the inside, and sometimes you'll have cells that are in between. So you have ectoderm, endoderm, and mesoderm. And it's from those different types of cells that we will start seeing our basic differentiation that's going to become things like our skin, our bones, or our organs. So it's important that this process is understood, and that it's so simple and elegant in its design, but it forms the basic preformation for our rest of our organs and our bodies and our body plans, not just for us, but for all animals in the animal kingdom. So then what is a protostome and what is a deuterostome? So essentially you're making the digestive tract. And for most species, there are two ends. There's a mouth and an anus. Well, when you start making that indention, what end does that indention become? Is it become your mouth, or does it become your butthole? Or anus. So for most species, it becomes their mouth, and they're called protostomes, meaning mouth first. So what about the species that don't have their mouth form first? We call those deuterostomes. In other words, that first indention becomes their butt, that becomes their anus. Excuse me for not using technical terms the entire time. I think it's kind of funny. So if your mouth forms first, you're a protostome. If your anus forms first, you're a deuterostome. Guess which one we are? You can probably tell by my intonation in my voice. Pretty much everything except for the echinoderms, like starfish, and the vertebrates, the chordates, anything that has a spinal cord and a backbone. All of everything else besides those two are protostomes. So like jellyfish, or not really jellyfish, let's go with like any type of the worms, flatworms, roundworms, segmented worms, any of the insects, or the arthropods, all the mollusks, all of those, their mouth forms first. With echinoderms, starfish, and vertebrates, us, our butt forms first. So if you want to think about it like this, for a little bit of point in time, you're nothing more than a butthole and a ball of cells. But that's how we all started. Some of you stayed that way, but the rest of us have moved on. We've all moved on. We're all doing good. We were all buttholes and a ball of cells for quite some time. That's the difference between protostomes and deuterostomes, and that major definition between those two is something we'll see and define for every group that we cover, because looking at patterns of development and how simple or complex their systems are generally evolves around protostomes and deuterostomes. So here's a good little picture of what we just kind of covered. So porifera and cnidarians don't really fall under the protostomes, simply because porifera don't have mouths or digestive tracts as we know them. Cnidarians, while they do have mouths, their mouth is also their anus, so they don't fall in. That indention never became two separate ends, so they don't fall to protostomes and deuterostomes. But platyhelminthes, annelids, mollusks, nematodes, arthropods, all of them are protostomes, where we have echinoderms and chordates are all deuterostomes. So all of these are mouth forms first, and us and our buddies the starfish are butt form first. Yay! So the last thing we're going to cover for this particular lecture is the architectural patterns of animals. And so essentially what does that mean? Is essentially what's their symmetry? How is their body plan derived, and what's the similarity between that and other organisms? Because if they have similar symmetry, then they generally have similar relations. So there's three basic ways you can have symmetry. You can either be asymmetrical, which means you don't really have symmetry. You can be radial, which means you're based off the concept of a circle. Now, some of them can have a specific type of radial symmetry called pentaradial, meaning 5 points around a circle. But radial symmetry is still the basics. And then we have bilateral symmetry, essentially meaning that if you cut it in half, you have even sides you have two even halves, kind of like mirror images of each other. Another term that we use is spherical symmetry, but we don't see this so much in the animal kingdom, but it does exist in things like some protists have a spherical symmetry. But since we're not covering protists, this doesn't fall under the topic of what we're talking about today, but there's an example of spherical symmetry. So the other big thing is going to be body cavities. Now this one's not a tough one, but it's weird to think of, and a lot of students seem to have problems getting these. We have holes and tubes that run through our body. The term for a hole is called a coelom. So something that is a coelomate has a body hole in it. Something that has an acoelomate doesn't have a body hole that goes through it. And something that's a pseudo-coelomate kind of looks like it has a body tube through it, but it's a false body tube because pseudo means false. So coelom means body cavity. We have the acoelomate, the pseudocoelomate, and the eucoelomate. The big difference between these is, again, with something with the acoelomate, there's no body cavity. There may be a tube running through it, and the tube is hollow, but there's no body cavity surrounding that tube. So think of like the digestive tract in a flatworm. The organ that makes up the digestive tract is straight up against hard tissue. There's no cavity for it to be in. Think of like our abdomen. Our abdomen has an organ sitting in it, and those organs can move around within that body cavity. It doesn't happen like that for an acoelomate. For a pseudocoelomate, they have a body cavity. They've got all this space around their organs, but there's no tissue connecting those organs back to the body cavity wall. And so we call these pseudocoelomates. So not only are the organs there and they can move around, but they can move around a lot and they get sloshed around and there's nothing really holding them in place. Whereas we have coelomates are also called eucoelomates, meaning true coeloms. We do have a body cavity, and there's also a layer of tissue that surrounds the internal organs that attaches to the outer body wall so they don't stay in place. When you turn real hard to one side, do your organs turn with you and stop, or do they turn and keep going? Well, for us, they turn and stop because we've got a bunch of connective tissue that holds them in relative position inside the body cavity. So acoelomates don't have a body cavity. Pseudocoelomates have a body cavity, but there's nothing to hold their organs in relative position. And then eucoelomates or just coelomates have a body cavity, but have connective tissue that holds their organs in place. And so the last bit of organization we see with animals, and increasing in complexity, this becomes more and more extremely common, is something called segmentation. It's a part of the specialization of their body plans. Segmentation means the body is broken into certain segments, and that segment handles certain operations of the body. Something like our cranial unit to our thorax to our abdomen. All of those are resemblances of a segmented body plan. When we cover segmented worms, we'll definitely see segmentation because the body plan of a worm is broken up into many different segments. Another big thing we'll see that's a common theme with animals is the concept of cephalization. Cephala means head, and not all species of animals have heads or even have segmentation. Some of them they don't have a distinct head or a distinct tail. As animals got more complicated, it became kind of important to make sure that you had a head, and in that head is usually most of your sensory organs, and that head was at the front of the anterior part of the body. So that way it's in front of us because that's the side of our body that we first encounter the world with, so it makes sense to have our eyes, our nose, and our mouth on the front part of our body. That theme is common throughout much of the animal kingdom, but not all. Again, some animals don't show cephalization. They, like jellyfish, they don't have a distinct head. They don't have a distinct division of parts of their body. So segmentation and cephalization is a common theme we'll see in the animal kingdom, and that we'll use as a tool to help us study and divide up some of the groups into different phylogenies based on the presence or absence of these features.